You might be wondering where all this sponge bath came from. You might also be thinking that these sponges are simply for cleaning your dirty dishes in the kitchen or scrubbing the dirt out of your body. Well, yes, you're partly right. But there's still more with this simple animal which you have been using almost every day.
Robert Grant coined the term porifera. Pori means pores, fera means layers, commonly referred as sponges. A multicellular invertebrate animal that diverged from Proteus 600 million years ago, and not a whole lot has changed for them since then. Sponges are an ancient and highly successful group of animals, vary in size up to 1 meter high in shape. In the Paleozoic, they are believed to have comprised more than half the biomass in marine reefs. People often think of sponges as plants rather than being animals. This misconception is due to some of the characteristics of the porifera.
Like plants, they do not move. They stay put in one place, stuck to the bottom of the water, either salt or fresh. Also, they don't have mouth, muscles, chew tissues, and organs or organ systems. They are heterotrophs that are dependent to other organisms and cannot manufacture their own food. Sponges are sedentary, filter-feeding metazoan which has a single layer of flagellated cells that drive an unidirectional current of water through its body. Sponges occur in the rivers and streams, from rock pools to the deep ocean depths, from frozen Arctic seas to the warm tropical seas. They are perhaps at their most beautiful in tropical marine seas. There are about 10,000 known species, and though their basic organization is pretty simple and remains fairly constant throughout all species, they do manage to show a great variety of forms.
While sponges do not exhibit tissue layer organization, they do have different cell types that perform distinct functions.
Pinacocytes, which are epithelial-like cells from the outermost layer of sponges enclosing a jelly-like substance called mesohyl, which acts as an endoskeleton or internal support, maintaining the tubular morphology of sponges. Sponges have multiple pores called ostia on their bodies that allow water to enter the sponge. In some sponges, ostia are formed by porocytes, single tube-shaped cells that act as valves to regulate the flow of water into the spongicle or central cavity.
They can change their size or they are contractile and can therefore change the size of the openings of the ostia, thus controlling the flow of water through the sponge. Pinacocytes are also implicated in the absorption into the sponge of larger food items.
Choanocytes or collar cells are present at various locations, depending on the type of sponge. However, they always line the inner portions of some space through which water flows, whereas pinacocytes line the outside of the sponge. Choanocytes tend to line certain inner portions of the sponge body that surround the mesohyl. The structure of a choanocyte is critical to its function, which is to generate a water current through the sponge and to trap ingest food particles by phagocytosis.
Note that there is a similarity in appearance between the sponge choanocyte and choanoflagellates or protista. This similarity suggests that sponges and choanoflagellates are closely related and probably share a recent common ancestry.
The second crucial cells in sponges are called amoebocytes, named for the fact that they move throughout the mesohyl in an amoeba-like fashion. Amoebocytes have a variety of functions, delivering nutrients from choanocytes to other cells within the sponge, giving rise to oocyte, a female gametocyte, a cell that develops an egg for sexual reproduction, which remain in the mesohyl. Some of these more specific cell types include collencytes and leukocytes, which produce the collagen-like protein to maintain the mesohyl, sclerocytes, which produce spicules in some sponges, and spongocytes, which produce the protein spongin in the majority of sponges. These cells produce collagen to maintain the consistency of the mesohyl.
Some sponges have both types of skeleton which have spicules and spongin at the same time.
The water flowing through a sponge simultaneously serves as its respiratory, excretory, and internal transport system. As water passes through the body wall, sponge cells remove oxygen from it and give off carbon dioxide. Metabolic wastes produced by cellular respiratory, such as ammonia, are also released into the water, which carries them away. A sponge 10 centimeters in height and 1 centimeter in diameter was found to pump 22.5 liters of water per day through its body. The water that flows through the body of the sponge also plays a role in sexual reproduction. Although eggs are kept inside of the body wall of the sponge, sperm are released into the water flowing through the sponge and carried out into open water. If those sperm are taken by another sponge, they are picked up by amoebocytes to the sponge's eggs where fertilization occurs. The zygote that result develops into a larva that swims and can be carried by currents for a long distance before it settles down and grows into a new sponge. Sponges reproduce sexually as well as asexually. Faced with cold winters, some freshwater sponges produce structures called gemmules. Gemmules are spear-shaped collections of amoebocytes surrounded by a tough layer of spicules. Gemmules can survive long periods of freezing temperatures and drought, which would kill adult sponges. When conditions again become favorable, gemmules grow into a new sponges. Sponges can also reproduce asexually by budding. In this process, part of sponge simply falls off the growth into a new sponge. Their power of regeneration is remarkable. If you were to grind a sponge, separate its cells by filter, its cells would clump together and grow into several new sponges.
Despite of the simple structure of sponges, they play a huge role in cleaning up the ocean floor. Since the time of the Greeks and Romans, humans have used the dried and clean bodies of some sponges in baking. Most sponges you see in supermarkets today are artificial, but natural bath sponges are still available. Recently, scientists have found uses for parts of the sponge other than its skeleton. In a series of exciting new developments, scientists are learning to use several chemicals manufactured by sponges. Because sponges cannot move, they must protect themselves from their enemies. And in other ways, bacteria and many tiny organisms are constantly looking for surfaces on which to settle. To protect themselves from being overgrown by these organisms, sponges manufacture numerous compounds that are toxic to such organisms. These chemicals also discourage many from chewing on sponges. Researchers have found that many of these chemicals are powerful antibiotics that can be used to fight bacteria and fungi causing disease. Other sponge chemicals act against viruses almost as well as antibiotics that can be used to fight bacteria. One compound taken from a Caribbean sponge may be useful against leukemia. Another may help fight certain forms of arthritis. Still, other sponge chemicals may be effective against bacteria that cause strep throat and those that become resistant to penicillin. Although most of these drugs are still in the experimental stage, scientists hope that they will soon be ready for human use.